So now that you've watched the diagram videos pertaining to replication, transcription, and translation, it's time to talk about mutations and how they can affect those processes. So with a mutation, it's basically a change to the nucleotide sequence in DNA. So if the nucleotide sequence is meant to read ATGC, and it now reads ATGG, it has acquired a mutation which can ultimately impact the cell. Because remember, the DNA contains the gene regions or the recipes for how to produce a protein. And if a mutation occurs in the gene region, ultimately that is going to affect the production of a specific protein. And depending on what protein we're talking about, that basically impacts what may happen with the cell. So for bacterial cells, when they acquire mutations, it can mean a variety of different things. Sometimes they can acquire mutations that can ultimately lead to cell death, but sometimes they can acquire mutations that can change them in some way, sometimes even to their advantage. So sometimes these genetic mutations may cause them to look different. Maybe their shape changes or their size changes. It could cause them to develop different nutritional requirements. It could change their temperature preference from preferring a cold environment to maybe preferring a moderately warm environment. It could even change their susceptibility to antibiotics. So because of mutations, a bacterium could become resistant to an antibiotic such as penicillin. So understanding mutations and their impact on bacteria is quite important. There are different types of mutations that can happen within a cell. So the first being spontaneous mutations and the second being induced mutations. So spontaneous mutations happen as a result of errors in DNA replication. So by this I mean the DNA polymerase III enzyme, the enzyme that is responsible for synthesizing the majority of the DNA outside of replacing their primers. But this enzyme essentially synthesizes DNA, and it makes mistakes. So when it sees a T on the parent strand and it's supposed to put an A in the newly synthesized strand, let's say it accidentally puts a G there where there was supposed to be an A. Well, if it doesn't catch its mistake and fix it, and that mistake stays, that mistake is now a mutation. So essentially, spontaneous mutations are a result of errors in replication, where DNA polymerase III basically puts the wrong nucleotide in the wrong place. Again, it was supposed to put a T there, and it accidentally put a G there. And a lot of times, DNA polymerase III has proofreading abilities, so it is able to sometimes catch its mistake and then replace it. However, again, if it doesn't catch the mistake, if the mistake stays, that's where we get a mutation. There are also induced mutations. Induced mutations happen when a cell is exposed to what we call mutagens, which are things that are able to mutate DNA. This can be various different chemicals or even exposure to something like UV radiation. But what can happen is the DNA can mutate. It can either mutate extensively, which can lead to cell death, or again, it could mutate and change characteristics of the cell, potentially even making it stronger and more advantageous for the bacteria. So induced mutations are really as a result of exposure to mutagens, whereas spontaneous mutations just occur naturally due to errors in the replication process with DNA polymerase III. So regardless of whether the mutations happen through a spontaneous route or an induced route, there are different types of mutations. One being a point mutation, and the second being a frame shift mutation that we'll see next. So point mutations are mutations where one nucleotide is substituted for another. So to look at this, the original DNA code was supposed to read AAA. So when this DNA was put through transcription, the messenger RNA would have read UUU, which is a codon sequence for phenylalanine during translation. So this is if there was no mutations and everything occurred the way it was supposed to. But let's say there is a mutation, and now instead of the DNA code reading AAA, it now reads AAT. This right here is our point mutation. This was supposed to be an A, not a T. So unfortunately, when this gets transcribed and turned into RNA, it's going to say UUA now instead of UUU. And when the ribosome reads this codon sequence during translation, this codon sequence is no longer coding for phenylalanine. Instead, it's coding for the amino acid leucine. So that means where we were supposed to have phenylalanine built into that protein, now we have leucine, which could make the protein completely non-functional, or it could change the function of the protein. And again, depending on what that protein is being used for, that's going to kind of dictate what's going to happen to the cell. Sometimes mutations occur and it doesn't have any type of substantial impact. So to kind of look at that, let's say it was supposed to read AAA, but now it reads AAG. So right here, this is our point mutation. This was supposed to be an A and it's a G. So when this gets transcribed into RNA, instead of it saying UUU, now it says UUC. So when the ribosome reads this codon sequence during translation, fortunately, this codon sequence also codes for phenylalanine. Several of the amino acids have various codon sequences that code for them. So sometimes even when mutations occur, you can ultimately still get the same amino acid in the same spot. And to kind of look at that, we look at our chart and we can see how phenylalanine has 2 different codon sequences that code for it. Leucine has 6 different codon sequences that code for it. Serine has 4. So it kind of gives some flexibility with the mutations where you could still potentially get the right amino acid in the right spot even once mutations have occurred. A more serious type of mutation is going to be frame shift mutations. Frame shift mutations are when an extra nucleotide is either inserted or deleted from the DNA strand. So if the DNA strand, this is what it was supposed to originally read, so this is the non-mutated strand. So when this is taken through transcription, this is the messenger RNA that we would have produced. And then when the ribosome reads the codon sequences in the messenger RNA, this is the chain of amino acids that would have been built to build the protein. So this is if everything works the way it was supposed to work. So in a deletion, what's going to happen is an entire nucleotide is going to be deleted from the DNA sequence. So right here where we're supposed to have a G, we're actually going to get rid of it. So a deletion has occurred. So that means when this goes through transcription to make messenger RNA, we're going to be missing a C here. And so when now that we're missing a C, when the ribosome comes in to read the codon sequences, it's put on a completely different reading frame. Because you have to remember, the ribosome reads in those 3 nucleotide codon sequences. So if you remove one from the beginning, it messes with the entire reading frame throughout the rest of the messenger RNA. So you're getting a completely different sequence of amino acids as what you were supposed to get if it was non-mutated. And ultimately that means that the protein is not going to be made, which is ultimately going to affect the cell in some way. It's the same concept with an insertion, except you're adding in an extra nucleotide. So we have an extra C here where we're not supposed to have one, which means we're going to have an extra G when we take the DNA and transcribe it into messenger RNA. Again, since we're adding in an extra nucleotide, when that ribosome reads those codon sequences, that extra one, again, messes with the reading frame. So now it's reading a completely different series of codon sequences all the way down. You're going to get a completely different chain of amino acids, ultimately not building the protein that you're supposed to make within the cell, which is obviously going to negatively impact the cell. or with bacteria, it could potentially even positively impact them and make them stronger. So one kind of simple technique I use for this is if you know the code for the non-mutated DNA, and if you know the code for the mutated DNA, it's actually really simple to figure out what type of mutation happens. So if there's the same number of nucleotides between the non-mutated strand and the mutated strand, a point mutation probably happens somewhere in there, and you can kind of look to see where it may be different. But if it's the same number, it's probably a point mutation where one was just substituted for another. If there's one more nucleotide in the mutated strand, then it's probably an insertion, which is a frame shift mutation. If there's one less nucleotide in the mutated strand, then it's likely a deletion. So it's very easy if you just count how many nucleotides are in the original strand, and then count how many nucleotides are in the mutated one. If it's the same, probably it's a base point mutation. If it's one more, it's an insertion frame shift mutation. If there's one less, it's a deletion frame shift mutation.